Okay, let's take a look at your Chapter 18 study guide. Let's go ahead and review so you can make an A on this test coming up. All right, number 1. Know that scientists came up with scientific names using Latin names because there are regional differences that can cause confusion. An example, buzzard in the UK means hawk, while in the US it means vulture. Now, this is a statement, there's no question here, but there's some stuff that we can expand on. The reason they use Latin names is so two or more scientists working from different countries know what each other is talking about. For example, if we go to the UK and a US scientist goes to the UK and he starts talking about a buzzard, he knows he's talking about a vulture. In the UK, the scientist is going to think that that guy's talking about a hawk. They're talking about two different organisms. By using Latin names, they know what each other is talking about. Now, these Latin names are given to both living and extinct species. Number 2. So if we had an organism with the name Papio anubis, which part identifies the genus? Which part identifies the species? And to be useful, the scientific name has to identify a single species. And what term describes the two-part name? So when we're looking here, we have two words. We have the Papio and we have Anubis. Well, excuse me. The genus is always going to be the first word. It's always going to be read as genus followed by the species. And the first part of the word is always capitalized, which you see here, and always identifies the genus. So the second word is always lowercase and always identifies the species. And it's usually going to be done by a trait or part of its habitat. But here it says which part identifies the genus. Would that be the Papio or the Anubis? And it's going to be Papio. That is the genus. And the species is going to be right after the genus, and that's going to be the Anubis. Now, the species is usually going to be identified by a trait or part of the organism's habitat. And the two-part name, and it's always going to be two names, genus, then species, and that structure is called the binomial nomenclature. That is the structure that is used. They're going to list the genus name followed by the species name, and that is by meaning two names, binomial nomenclature. All right, number 3, know what is included in and who developed this taxonomical structure: the species, the genus, the family, the order, class, phylum, and kingdom. Well, that is Carolus Linnaeus. He developed the taxonomical structure, and there are 1, 2, 3, 4, 5, 6. There are 7 taxa. Now, the genus is a group of similar species. So if you take all a species and you group them together, you're going to get a genus. You can take the genus, group them together, and put them into a family. You can take the families that are similar, group them into an order. You can take similar orders and you can group them into a class. You can take similar classes, group them into a phylum. You can take similar phylums and group them into a kingdom. And this is a visual representation. So we take species, we can put them into a genus. We take similar genera together, we can put them into a family. Take similar families, we can put them into an order. Here we take the similar orders, we put them into a class. We take all these similar classes, we can put them into a phylum. We can take everything in the phylum, group the similarities together, and put them into a kingdom. So goes from this order, going from the bottom to the top, is from the most specific to most broad, going from the top to the bottom, the most broad to the most specific. Number 4. What traditional, and when we say traditional, we're talking about science back then. What system did they use to classify organisms, and what do scientists use today to classify organisms? Scientists used to look for visual similarities, and today they use DNA to classify organisms into the appropriate clade. Now, this needs to have a D on the end here, and what scientists today use to classify organisms is the DNA. Number 5. Here we have a statement, there's no question. You need to know that all species in one genus should be closely related to each other than they are to any other genera, which is plural for genus. All right, and that's what we have here. So the species should be more closely related to each other rather than any other part in the genus. So for example, this bear right here, if we said it's a polar bear, they should be more closely related to polar bears than they should grizzly or black bears, whatever that may be. All right, number 6. What is a derived character? It's an inherent trait that gets passed on from a common ancestor. That's all it is. And the common ancestor is the key word here. All right, so here, if we take a look at this, the vertebral column, this is a derived character, and all these organisms have it, with the exception of the lancelet. Here we have hinged jaws, which is a derived character. Everything above the hinged jaws has it, so that would be the tuna, the salamander, turtle, leopard. Here, 4 walking legs would be a derived character of the salamander, turtle, leopard. The amniotic egg of the turtle and the leopard. And the hair, that's a derived character for the leopard. Number 7, what does similar DNA sequences show evidence for? Well, because we all have the same DNA ingredients, if you will, it shows that we come from a common ancestor. The DNA contains 4 ingredients, A, C, G, and T, and that is the adenine, cytosine, guanine, and thymine. Number 8, how do scientists find out how closely related 2 dissimilar organisms are? For example, cow and yeast, they are nothing alike, but if things come from a common ancestor, we have to figure out how closely related they are, and even though they're not closely related, we can still figure out an approximate range. Well, they look at the DNA to see how similar they are. You can look at the DNA because it has the same 4 ingredients. Here's mitochondrial DNA codes to see how these things are related, and you look at the letters and we can see the red panda, the giant panda, the American black bear, and the raccoon have the similar codons. They have ATT, 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 ATC. So very, very, very similar here. And the same thing with the rest of the codons. They're not exactly the same, and they can't be exactly the same because we have different species here. Those minor differences make up the different animals. Number 9, know the cell types for each of the 6 kingdoms. Well, you have eubacteria and archaebacteria. Those are prokaryotes. And if they're prokaryotic, we know they don't contain a nucleus. That's 2 of the 6 kingdoms. The other 4 kingdoms are protista, fungi, plantain, and mammalia. And those are eukaryotes, and they have a nucleus. Now, what is going to throw some students off is this eubacteria. They're going to see the EU and say, eubacteria, they're eukaryotic. No, they're not. Don't forget this key word, bacteria. All bacteria are going to be prokaryotes. So don't just look at the EU, look at the actual word bacteria. When you see bacteria, you know they're going to be prokaryotes. Down here, we don't see bacteria, so we know they're going to be eukaryotes. Don't let this pro and protista throw you off thinking it's prokaryotic. You need to spend time with this, familiarizing yourself with the fact that protista are eukaryotes and that eubacteria and archaebacteria are prokaryotes. We can see that right here. You also need to know that eubacteria and archaebacteria are unicellular. They're all unicellular. Now, the protista, some can be unicellular, but some can also be multicellular. Same thing here with the fungi. And you can see the same thing here in the plantae, and then all animals are multicellular. But our archaebacteria and the eubacteria right here in those kingdoms, they are unicellular, and they're the only ones that are only unicellular. All right, moving on, number 10. Why is protista written in quotes and not considered true clade? It is more closely related to organisms of other groups than the organisms in their own group. So the group that they're in, they're more related to ones outside their group than the ones in their own group. You can not put all of its true descendants and common ancestors into a single clade. And we came up with this example in an earlier class. Someone had a good question about an analogy of what this means. So think about if you have a brother or sister, what this means, what this is saying is you're actually more related to somebody you don't even know than your brother or sister. And that's why it's not a true clade. And if you're in a group with a family, you should be related to those more than people in another group. And that's why it's not a true clade. You can't put all of its true descendants and that common ancestor into a single clade. Number 11, why did scientists come up with the 3-domain system? Well, after scientists scrapped the Monera group, they came up with eubacteria and archaebacteria. Now, in the 1990s, which wasn't even that long ago, they found that these 2 kingdoms were more dissimilar than they originally thought. And that led to a new taxonomic level, the 3 domains. So you can see that we have these 7 kingdoms. There's 6 kingdoms. There are 7 taxa systems, 6 kingdoms, 1, 2, 3, 4, 5, 6. And then there are 3 domains: the bacteria, the archaea, and the eukarya. So the archaea refers to the kingdom of the archaebacteria. Bacteria refers to the kingdom of the eubacteria. Eukarya, which means eukaryotic, the eukaryotes refers to protista, fungi, plantae, and animalia. So there are 6 kingdoms right here. There are 7 taxa, which are the species, the genus, the family, the order, the kingdoms, the phylum, the class, all those that we looked at earlier. Cladistic analysis considers derived characteristics that have arisen as lineages have evolved over time. That's a statement, no question. This is, you can use a cladogram for cladistic analysis. You need to know that when you analyze a cladogram and you're looking at derived characteristics that have arisen as lineages have evolved, that is called cladistic analysis. Last one, number 13, DNA analysis can supply further evidence of relatedness. So in general, the more derived genetic characters two organisms share, the more closely related they are. And we saw this with doing clades. So here we have a statement, no question. And you can see that the DNA analysis, if we look at the DNA of a turtle and a leopard, that they're going to be pretty closely related. So it's not just the cladistic analysis looking at those inherited characteristics, but also looking at the DNA. So that's going to be it for chapter 18. If you have any questions, text me through Remind, let me know, and we'll make sure we cover it. We'll see you guys tomorrow.